Hi there, and welcome to LambdaU. My name is Darren, and I am a Solutions and Support Scientist at LICOR. In this section, we will describe the science and best practices behind sample preparation for your Western blot. Your Western blot can only be as good as the quality of the samples that you start with, so your attention to detail now will be rewarded later. Remember, proteins are sensitive, so treat them with care, and you'll be rewarded with high quality, accurate, and precise data. Prior to immunodetection, proteins must be isolated from their source, typically cells or tissues. In order to ensure a reproducible and accurate Western blot, extracted proteins must be stabilized in order to reduce protein degradation or loss of post-translational modifications. Protein samples can be isolated from tissues, cells, or even viruses. When your sample is coming from cell culture plates, ensure that the cells are washed with cold buffer in order to remove any excess salts or excess serum that can further contaminate your protein samples. If you're isolating proteins from tissues, it's recommended to discard the lipid layer following centrifugation. Commonly, proteins are isolated from either cells or tissues through disruption of the cell membrane, known as cell lysis. Treatment with mild detergents like NP40 or Triton X100 break down the cell membrane and release cytosolic components. More stringent buffers, like radioimmunoprecipitation assay buffer or RIPA buffer, release nuclear components and dissociate protein-protein complexes. Choosing the appropriate lysis buffer will depend on your target, its localization, and your downstream applications. Lysis buffers also contain compounds that stabilize proteins following cell lysis. The use of protease inhibitors and working on ice can reduce enzyme breakdown of proteins known as proteolysis. If you're studying post-translational modifications, it will be critical to add phosphatase inhibitors like sodium orthovanadate. Even with the use of protease and phosphatase inhibitors, it is helpful to limit protein degradation by working at low temperatures. After cell lysis, protein should be kept on ice or stored at -80 degrees Celsius for long-term storage. We also recommend storing your samples in small aliquots to avoid freeze-thaw cycles. Now that we've learned how to isolate and store our proteins, we can learn how to determine how much protein we have and how to prepare our samples for downstream applications. We'll talk more about that in the next lesson.